So, our first big process that we're going to deal with in Unit 3 is DNA replication. Unit 3 actually deals with quite a few processes. So, our first one is DNA replication. We also cover mitosis and meiosis. We do protein synthesis, which I think is all we're going to cover in this lecture. But later on, you guys do some info on biotechnology practices as well that you need to know the steps for. So it's a whole lot of memorizing things and learning very, very lengthy processes. So DNA replication, I like to think of, has four different characters. Which are all different types of enzymes. The first one that we have is DNA helicase. The second that we have is DNA polymerase, which are our two most important ones. And these two are the only two that you really need to know a great detail for in the syllabus. The other ones that we have are primase and ligase. Now, while these two are not in the syllabus explicitly, I have seen multiple choice questions come up that deal with the roles of primase and ligase. So it's still important for you to know something about them. I don't think you need to explain their roles per se, but you need to know the process in terms of these four in these four enzymes. So our first step, we have our DNA double helix, kind of looks like this, right? Something like this. And helicase, which is our red enzyme, will come in and unzip the DNA helix, the DNA double helix, by breaking the hydrogen bonds, exposing the nitrogenous bases. So, as you can imagine, if those hydrogen bonds are broken between adenine and thymine and cytosine and guanine, we've got all of these bases which are now freely exposed to organelles, enzymes, ribosomes, anything that wants to do anything to them. So this is the way that your cell can access information that's coded within your DNA. Helicase will then continue to unwrap the DNA strand until it gets to the end of the gene. And then we move on to step 2. So step 2 starts to happen while helicase is unzipping, by the way. This is all happening at once. Primase will lay down RNA premise to the strands that are being copied. So essentially, if we're doing DNA replication, we want to have one DNA strand be copied into two identical DNA strands. So if this is my original strand of DNA, like so, and my helicase is continuing to unzip it, there's helicase there. Oh, I'm just gonna write all of the bases in like so. Primase will come in. What color did I make primase? Here we go. And basically lay down a primer at certain spots on the DNA strand that needs to be replicated. Kind of like if you were putting on primer before painting a wall, it's something that helps the paint adhere to the wall and get stuck on there properly. Or if you're doing like primer before putting on makeup, it's something that helps all of your makeup stick onto your face and stay. The primer for laid down by primase helps the copied DNA strand or the extra copy that we're going to make adhere onto the original copy more efficiently. So it's essentially like a priming step that helps the copied DNA strand stick onto the original DNA strand better. So now we go into step 3. Then comes in our DNA polymerase. DNA polymerase is also another enzyme, and it lays down complementary base pairs to the original or template strand. Pairs to the original strand. So this time I'm going to make this a little bit bigger, like so, like that. So imagine this is our 5' end of the original strand, and this is our 3' end. Remember, that's the end that the sugar is attached to, right? And we start off with a couple base pairs. Um, like so. And so we've got A, T, oh, oops. We've got A, T, T, A, G up here. So that means we have T, A, T, C on the top. What will happen is DNA polymerase will start coming in. So this is our polymerase, and joining up complementary base pairs to the template strand. So it'll say, okay, if this is a T, you get an A. This is an A, we get a T. We then get a T, another A, and then a G, and starts joining up all of the complementary nucleotides in one direction. However, the catch is that polymerase can only add bases or operate in the 5' to 3' direction. So here, if we've got our 3' end, it's starting off in the 5' end, right? And then moving this way. That's the only way that polymerase can continue working. So on this top strand, polymerase is all right to just continue, oh, whoops, sorry guys, continue working until the end of the strand and copy the same direction that helicase is unraveling in. However, for the bottom strand, technically this starting point is the 3' end. Unfortunately, DNA polymerase cannot work in the opposite end, the 3' to 5' end, so it has to start somewhere random, essentially. So this is our polymerase molecule here. Sorry, wait. I'll make it start where I ended it before. So this is where polymerase will need to start, and then it can continue adding on the nucleotide base pairs in this order. So C, T, A, T. And then once it gets to the end of the strand, it has to jump back to a position it hasn't been to and continue replicating until it gets to the base pair before the base pair that it started on the previous time. So it would jump back until here and then continue building the strand until it got to this point. So because of this jumping back motion, we end up with a strand that has continuous replication, so it just follows the same direction as helicase, called the leading strand. And then a strand that has discontinuous replication or replication in certain chunks, called the lagging strand. However, polymerase will continue to add the nucleotide base pairs until we end up with two template strands that are completely separated from each other. So again, we've got 3 prime, 5 prime, 5 prime, 3 prime, and all the base pairs. Mixed with 2 completely copied or new strands fixed by DNA polymerase. Like so. The only problem is this strand on the bottom here will not be completely connected together. It'll be sort of in chunks. So as you can see here, there'll be some spaces between them. So this chunk will be separate depending on where DNA polymerase had to stop and start. This is where our final enzyme comes in. Here, this is ligase. Ligase is sort of like a glue enzyme. So ligase will glue all the fragments together, producing 2 identical strands of DNA. These individual fragments caused in the lagging strand are also called Okazaki fragments as well. That has come up on not a QCEE multiple choice question, but it came up in like a mock exam by a different company. So I'm just going to teach you guys, but they are called Okazaki fragments. Ligase's job is to come in and glue all of them together. And then finally, we end up with, hopefully this isn't over my video too much, but one of the DNA strands will be completely put together, and the other DNA strand will also be completely put together, and they will curve up into the double helix. So I've got a nice diagram of that on the next slide to show all of you. Here, this is a cleaner diagram, but it doesn't explain all of the steps. We've got helicase on this side. Wait, let me get the laser pointer. There we go. Helicase on this side unwrapping the DNA, breaks hydrogen bonds, exposes bases. That's what you've got to know. Then we've got primase coming down and laying down the RNA primers. We then have DNA polymerase, which is working to put all of the complementary base pairs together. You can see on the 3' end of the template strand, the 5' end of the copied strand is just being continuously made. And then on the 3' end, DNA polymerase is starting here and moving in the opposite direction. So that we get these little Okazaki fragments happening. DNA ligase then joins the Okazaki fragments together. So you end up with 2 parallel strands of DNA, and then they end up wrapping around each other to form 2 double helixes at the very end. Alrighty, so that's the whole process of replication, right? And replication is only one part of the cell cycle, which we're going to talk about now. And when we're talking about the cell cycle, there are 4 stages. Replication happens in the S stage, happens in the S stage, and it doesn't happen at random times. The S stage is essentially a preparation stage for cell division. Cell cycle kind of has 4 main stages. G1, which we see here, is the growth stage. So this is just where your cell is growing, maintaining itself, doing the function that it needs to do. And then it'll get to a point where it realizes that the organism is growing along with the cell and it needs to divide so that the organism can grow properly. Because obviously cell division makes more cells, so the organism gets bigger. Once the cell realizes that it needs to divide, it moves from the G1 phase, or the just stock standard growth phase, to the DNA synthesis phase. In this, DNA replication will occur. So you've got two copies of the DNA in the nucleus now. So the nucleus can divide into two daughter cells. Then we've got G2 phase, which is growth and preparation for mitosis. Luckily for you guys, you don't need to know any detail about the G2 stage at all. I learned all of that in uni. It is a pain. But basically, it's just where all of the checkboxes for mitosis will occur. And finally, the M phase is actually where the cell division happens. The nucleus splits, the cytoplasm splits, and you end up with two identical daughter cells from one parent cell. And that, oh, oops. And that process of mitosis is basically what we're going to talk about now. So, in your own time, I hope that you guys can pause and try some of these questions just to consolidate your understanding about DNA, because it's very important that you're able to comprehend the information as well as absorb it. So have a go at doing those. But because it is a recording, I'm going to continue on with content. Now we're going to talk a little bit about cell variation and cell replication. So this is splitting of cells, cell division, and there are two types, mitosis and meiosis. Before we go on to talking about the actual processes behind those two though, I've got to talk a little bit about some basic knowledge. So first of all, we've got two types of cells, or two kind of categories of cells in our human body. The first one being somatic cells. So these ones are your regular body cells. They have no involvement in producing reproductive cells, and they've got nothing to do with fertilization and creation of gametes and zygotes and things like that. Germline cells, however, are the cells that end up producing sperm and eggs. So these two are the ones that fuse together to produce a baby in fertilization, basically. Diploid cells basically mean a cell that contains two sets of chromosomes. So all somatic cells have two sets of chromosomes, and we'll talk a little bit more about that in a later slide. Haploid cells, which are really only important for meiosis, refer to a cell only having one set of chromosomes, for example, the sperm and the egg cells. The reason being, sperm comes from your father, egg comes from your mother, and when the two of them fuse, you end up with a diploid cell, so the baby can have diploid cells. So now we go into our two types of cell division. Mitosis is where a parent cell will divide into two genetically identical daughter cells. And then meiosis is a parent germ cell that divides into four genetically unique. Oh, no, that should be genetically unique. Haploid cells. So we're going to talk a little bit about that process very soon. So, in the last, in the earlier part of the lesson, we discussed how chromosomes are formed by condensing DNA wrapped around histone proteins, and we've got all of these. We've got 23 pairs of chromosomes within one cell, within one human cell. Every single one of our cells, no matter if it's a nerve cell, a muscle cell, a stem cell, a liver cell, will have this identical set of 23 chromosomes included. When you're doing DNA profiling, you'll end up with this structure or this diagram called a karyotype. And it's basically a layout of all of the chromosomes found within a particular organism or individual in this case, and ordered from the largest chromosome, which is usually chromosome number 1, all the way to the sex chromosomes, which are chromosome number 23. We have 22 autosomal chromosomes, so that means not relating to the sex chromosomes at all. And then one set of sex chromosomes, which determines whether you have male genitalia or female genitalia, or reproductive organs, I should say. XX is typically for male reproductive features, and XY is typically for female reproductive features. Later on in the unit, we're not going to cover it in this lesson, but you will be learning about sex chromosome mutations as well. So how intersex individuals can originate and sort of what health implications you can have from there as well. So we've got each species will actually have its own set of chromosomes. In most humans, it is 26. However, chromosome mutations can cause different numbers of that to occur. And then when you're setting it all out in a karyotype, it's a paired set of 23 chromosomes. Our gametes are different in that they only have one half of the pair of chromosomes, which means that they only get 23. So later when we do meiosis and we have our egg cell and our sperm cell, each of those individual sex cells will only have 23 chromosomes. Um, and so now I just want to go through the structure of a chromosome a little bit more because this also came up as a exam question in my year and it tricked people up a little bit. We have one chromosome here, and chromosomes in cell division will contain basically two copies of each other after the whole replication thing happens. So when they split, you end up with identical copies of each of the daughter cells. We've got two arms. Typically a chromosome will have a short arm and a long arm, and a centromere at the center which holds the two sister chromatids together. At the ends, we have these structures called telomeres, which are also made of DNA. They don't code for anything, they just stop the chromosome from unraveling and they help them stay in this like X shape. And then across the chromosomes, we've got all of these genes. And basically, the gene for one particular the gene for a particular trait on one chromosome will be found in the same location in the other chromosome. So on these sister chromatids, we've got this could be like the gene for eye color. This is also the gene for eye color. This could be the gene for height. This could also be the gene for height, as examples. In reality, it is a lot more complicated than that, but typically speaking, genes are found on the same position and they are called the gene loci.